Lab 2 Measurement by Emma and Teddy.
Alright, here's some background information on gel electrophoresis. It is a technique in which the charged macromolecules are separated by their migration in an electrical field using a gel as a platform. Scientists use it to determine the size, mass, and charge of molecules such as proteins and nucleic acids. These are the components of electrophoresis needed to conduct the experiment. Agarose, which is a gel that provides a limiting matrix with small pores for the DNA to travel through. And what this means is small particles are going to migrate faster and large particles are going to migrate slower. TAE, a buffer system, is needed to maintain the constant pH. DNA loading dye, it causes the DNA to sink into the wells and adds colors to samples so they are easily visible. An electrical current, which provides the force that causes the particles to develop an attraction, positive or negative, and to migrate. And that will ultimately determine whether the sample is positive or negative.
Okay, our hypothesis is that molecules with the lowest mass will migrate faster through the agarose mixture than molecules with the largest mass under the charge of the electric current.
Step 1, preparing the agarose gel. The agarose concentration that we're going to use in our experiment was 1 times concentrate. We're going to make 30 milliliters of this 1 times concentrate agarose solution. The solution percentages are a function. Mass over the substance in grams over the total volume of solution in milliliters. This function is displayed below. Continuing on in step 1, we turned on the balance and waited until the readout showed 0.00 grams. Centered our Weibo on the balance and pressed the 0 button to tare it, allowing only the object of interest to be weighed, in this case, the agarose. We placed the agarose in the center of the Weibo and recorded the weight.
Here is a picture of our balance with the agarose, which weighed out to 0.30 grams. And this is us adding 0.30 grams of agarose to the flask.
Step 2, preparing the running buffer. 1 times concentrate of TAE would be used as the running buffer, not the stock solution of TAA in our lab, which was 50 times concentrate. So we needed to dilute it. We calculated the amount of TAA needed using the equation below.
As you can see, we used 8 ml of the TAE solution. We used 392 ml of deionized water and mixed them together to have a total volume of 400 ml. This is a photo of our graduated cylinder with 380 ml of deionized water.
Continuing on in step 2, we poured 380 milliliters of water into a large beaker and 12 milliliters of water into a smaller beaker. We poured all 392 milliliters of deionized water from the beakers into the container with the cap. Using a pipette, we added 8 milliliters of TAE to the 392 milliliters of water and then mixed the solution. This solution would later be used to dilute the agarose.
Here is a photo of us measuring out 8 ml of TAE solution to add to the 392 ml of water.
Here is our container of 1 times concentrated TAE, which is composed of 392 milliliters of deionized water and 8 milliliters of agarose. Step 3 was setting up the electrophoresis casting tray. We added 30 milliliters of TAE solution to the flask filled with agarose. We heated the agarose solution in the microwave for approximately 30 seconds. We removed the solution from the microwave and swirled it to make sure that all the agarose was completely dissolved. We then placed the metal spacers in the electrophoresis casting tray while we waited for our agarose solution to cool down. Step 4, pouring the gel. After placing the comb in the middle of the grooves of the casting tray, we carefully poured the agarose solution into the tray. We waited until the agarose solidified to a gel before we removed the metal spacers and comb. This is a picture of our casting tray with the solidifying agarose solution, metal spacers, and comb. Step 4. After gently removing the metal spacers and comb, we added the 1 times concentrated TAE buffer to the running chamber. This is us adding our 1 times concentrated TAE solution to both sides of the running chamber. For step 5, we prepared our samples. The first 4 dyes come as a 1 times concentrated stock solution, so we diluted each to a 0.25 times concentrated solution in a final volume of 20 microliters, using the equations below. Loading dye comes prepared as a 5 times stock concentration stock solution, so we diluted the loading dye to a 1 times concentrated final concentration in 20 microliters.
Using the equation mass 1 times volume 1 is equal to mass 2 times volume 2, we figured that our first 4 dyes containing a 1 times stock solution would contain 5 microliters of dye and 15 microliters of deionized water. The loading dye sample would contain 4 microliters of dye and 16 ml of water.
Continuing on in step 5, we used a micropipette to measure out the correct amount of microliters each solution should have. We placed a new tip on the end of our micropipette before we measured out each dye. This would ensure that none of the samples are contaminated. We followed these rules when using our micropipette. These rules are outlined below. Okay, right now I'm actually adding a new tip to our micropipette, and I'm going to withdraw 5 microliters, of this sample, which is with this dye, and we're going to withdraw it from its capsule. Now I'm going to place that dye that's contained in the micropipette into its respective capsule.
Then we correctly disposed of the micropipette tip into the hazardous waste container.
We're now on step 6, which is loading our samples. After preparing our 5 samples, we carefully expel them into the slots made by the comb in the agarose gel, which are also known as the wells. We placed a new tip on the end of our micropipette before we loaded and expelled each dye. Again, this is just to ensure that none of the samples become contaminated. Okay. This is us loading safranin into one of the loading wells in our agarose gel. Again, being careful to change each micropipette tip after ejecting each solution into the well, because contamination of a solution would cause the migration patterns of each sample to be different. Step 7, running the gel. Once all the samples have been loaded into the wells, we place the cover over the electrophoresis apparatus. Then, we attach the electrodes to the electrophoresis chamber and then to the power supply. We turn on the power to 150 volts for about 17 minutes. We turned off the power supply, unplugged all the electrodes, and, with gloved hands, removed the gel from the electrophoresis to visualize the gel.
This is a picture of us running the gel at 150 volts.
Alright, this is step 8, and after the 17 minutes of 150 volts being added to the agarose gel, we turned off the power supply, and then we measured the migration of the samples, which were xylene cyanol, orange G, bromophenol blue, 4-dye, and safranin, and we measured them using millimeters. Okay, these are our results, and xylene cyanol, bromophenol blue, orange G, and 4-dye all migrated towards the positive electrode, and safranin O migrated towards the negative electrode.
Xylene migrated a distance of 37 millimeters at a rate of 2.18 millimeters per minute. Bromophenol blue migrated 58 millimeters at a rate of 3.41 millimeters per minute. Orange G migrated 70 millimeters at a rate of 4.12. The 4-mix dye migrated 68 millimeters at a rate of 4. And safranin O migrated 39 millimeters at a rate of 2.29 millimeters per minute.
This is our conclusion. The xylene cyanol had the greatest molecular weight. It migrated the least distance during the 17 minutes of the gel running. Its molecules were too large to easily pass through the agarose matrix, which was composed of small pores, thus encountering a lot of resistance while trying to travel to the positive anode. Orange G had the lowest molecular weight. It migrated the farthest and fastest during the 17 minutes of the gel running. Its molecules were tiny enough to easily pass through the pores that the agarose matrix formed in the gel. Continuing on with our conclusion, we can see that our positively charged dyes was safranin O because it moved towards the negative anode, and because opposites attract, we would conclude that safranin O was a positively charged dye. On the other hand, our negatively charged dyes would be xylene cyanol, orange G, bromophenol blue, and 4 dye.